All right, hi everyone. Welcome back to Module 3, working with fMRI data. We're going to be continuing Section 1, the biological basis of fMRI, by talking about visualizing brain activity in this lesson. So we already covered brain activity. We're on Lesson 2 now, and then our future lessons will be about interpreting fMRI and applications of fMRI. So from our last lesson, we learned that brain activity is correlated with an increased blood flow due to need for oxygen, and that blood oxygenation level changes with varying blood flow and is measured by the Bold signal. So some questions we'll answer today are: what is brain imaging? What is an MR signal and how does it relate to fMRI? And what do different visualizations of the brain look like? So starting with brain imaging, brain imaging is a means of exploring the overall structure and function of the brain. So not only visualizing specific brain regions, but also the connections and activity between them. So this is used to locate brain damage, diagnose diseases, record metabolic activity, and understand the brain and its functional activities, which is what fMRI focuses on. So here are some different forms of brain imaging. EEG measures electrical activity in the brain, and this is kind of what the signal looks like from that. EMG measures the health of motor neuron paths to muscles, so also more of a electrical signal than a visualization of the brain. MRI measures tissue response to magnetic fields, so this is more of the typical view of the brain that you guys might be used to seeing. fMRI, which is what we're focusing on in this module, measures changes in blood flow to active brain regions, which gives us the colors on these pictures indicating the functional areas of the brain. There's also MEG, which measures magnetic fields in the brain, PET scans, which measure metabolic activity using radioactive tracers, CT scans, which are combined X-ray images, and kind of a newer form of brain imaging called optogenetics, which is the activation of individual neurons using really, really thin or really focused lights. So here it's like the most, precise view because we can see the individual neurons versus these larger scale brain images. So one of our questions was, what is an MR signal? So MR stands for magnetic resonance signal, and we're going to start from MRI. So I kind of want to explain how this relates to MRI first and how an MRI works, and then we'll get to fMRI and relate this to the Bold signal later. So an MRI, which is just magnetic resonance imaging, detects magnetic resonance signals produced by displaced radio waves in magnetic fields. So the hydrogen atom, which is something that is in water molecules, right, which is all throughout our body, hydrogen atoms behave like a small magnet. So the magnetic field in an MRI machine temporarily realigns the water molecules, which contain hydrogen atoms, right, H2O, in the body when you're in the MRI machine. And so those radio frequency waves cause atoms to produce magnetic resonance signals, which are detected by RF coils in the MRI. So here's kind of a better timeline of how this works. So basically in an MRI, there's two magnets that produce two different magnetic fields. So in this kind of chronological order, the first magnet is turned on, which causes the water molecules in the body to all align in one direction due to that sensitive hydrogen, that sensitive proton. And then a second magnetic field is pulsed on and off, which causes each hydrogen atom to change alignment when the second magnetic field is on, and then relax when off. So when I say change alignment and then relax, it's like if you think about if it's like a straight line, the magnetic field is going to cause it to go a certain direction, and then when it relaxes, it's going to kind of go in like a circle and relax back to its ground state. And so when electricity is passed through the gradient coil, it creates a magnetic field which causes those atoms to vibrate and produces unique signals that can be recorded. And those signals are going to be different. So those hydrogen atoms are going to relax in different ways depending on the tissue that it makes up. So that's kind of what creates that like distinction in those images. So how does this relate to fMRI? About 20 years ago, it was discovered that MR signals are not just sensitive to the different tissues in the brain, but also to brain activity. So when an area of the brain is active, the MR signal actually increases by about 1%, which is a very, very, very slight difference. But you can analyze that pattern of MR signal changes to reveal brain function. So you can see here, this is like a time scale of different signal changes in the brain that can be correlated to, or that can be kind of translated into an fMRI image. So recall the magnetic properties of brain function. Remember that the magnetic difference between oxygenated blood or oxygenated hemoglobin and deoxygenated hemoglobin affects the MR signal. So oxygenated blood produces a stronger MR signal, and we can kind of interpret that stronger intensity as like a brighter image, thus like changing this kind of color scale on the fMRI image. And this MR signal that I've been talking about for MRI, when translated to fMRI, is the bold signal, the blood oxygen level dependent signal. So that's kind of where it ties together, I suppose. So another thing we talked about was just how do you visualize the brain? There's a lot of different ways to visualize it. fMRI is definitely not the only way. This is one really cool video done by Neuroscape, which is a lab at UCSF, and they do some really cool research. But basically, it's depicting the electrical activity in the brain. So the 3D model of the brain seen here is derived from MRI data, and then the electrical activity in the brain is gathered from EEG data, which is something we'll talk about in a later module. So those like golden fibers that you see in the image are myelinated groups of nerve axons called white matter. And these axon bundles basically coordinate communication between different areas in the brain. And then you can also see there's like different signals that are passing through those axon bundles, like kind of like pulses of light in the video. And so there's a lot of different colors. These represent different frequency bands in this specific visualization. So there are theta waves, which are shown in red, and that's a low frequency band, usually around 4 to 7 hertz, that is usually observed in like drowsiness or meditation. There are, if you can see the green pulses of light, those are beta frequency bands, low to mid range, so 16 to 31 hertz, associated with a variety of mental states and physical activity. So beta waves might be reduced during very active movements and then increased during anxious thinking. And then there's also alpha waves, which are shown in blue. The video's done, but kind of around the edges is another low frequency band that is usually associated with relaxation or closing the eyes. So earlier, if you saw, there were kind of some color mixtures maybe, so those show activity in more than one band. And then something that wasn't shown here are gamma waves. These are the highest frequency brain waves we've been able to record, usually anything above 35 hertz, and they're associated with like very focused states and information processing in the brain. So this is just kind of like a fun, different visualization of the brain, and it really shows that you can combine different brain imaging techniques to produce a much more advanced image of the brain in general. And there's also this kind of software. I've linked it here. If you click on it here, it's like the BrainNet Viewer. It kind of like shows different, or it's like a software that lets you play around with different visualizations. So for example, you can do brain surface only. You can do like volume to surface mapping. You can do brain surface for ROI construction. This last one, kind of regions of interest. Just kind of a cool tool that is available online, and I think created by some researchers in the lab. And then lastly, just fMRI. I'm sure you guys have seen images like this all the time, or videos going through the slices of the brain and kind of revealing where the functional activity is happening during whatever stimulation is going on. So also this little graphic down here kind of relates that bold signal change to different levels of activity that kind of associate with brain function. So just kind of looking at some different visualizations, especially since we're focusing on brain imaging here. But yeah, this lesson, basically to summarize, this lesson and the last one kind of just remember that oxygenated and deoxygenated blood have different magnetic properties. Specifically, oxygenated blood has a stronger signal than deoxygenated. And also the displacement of the waves that produce an MR signal can be detected by the MRI or fMRI machine. And those changes in those signals reveal the pattern of brain activity that we're interested in. So what's next? The next lesson is going to be interpreting fMRI. So we're going to dive deeper into the bold signal, talk about spatial versus temporal resolution, and then touch on some advantages and disadvantages of fMRI. Thank you guys. See you in the next video.